Pro-Vice-Chancellor, Campus Principal, Professor Rosemary Bell Antoine, Deputy Principal, Professor Indar Ramnarayan, featured speaker, Professor of Microbiology and Biotechnology, Professor Adesh Ramsubhag, Dean, Faculty of Science and Technology, Dr. Brian Coburn, other deans and heads of department, and given the occasion, I'd like to recognize UWI professors in the audience, members of the private and public sector, friends and family of Professor Ramsubhag, specially invited guests, members of the Open Campus Committee, special members of the media. Pleasant good evening to everyone. On behalf of the Open Lectures Committee of the St. Augustine Campus of the University of the West Indies, the host of today's event, I extend a very warm welcome to everyone. My name is Derek Chady, Chair of the Open Lectures Committee, and it's my honor to chair today's Professorial Inaugural Lecture by our well-respected Professor Adesh Ramsubhag, Professor of Microbiology and Biotechnology. A Professorial Inaugural Lecture is a momentous and esteemed occasion to recognize and celebrate the appointment of an outstanding member of the university community to the rank of professorship. At the University of the West Indies, professorship is the highest academic rank, and the Open Lectures Committee celebrates the outstanding achievements of Professor Adesh Ramsubhag. Professors play a vital role in the university community. They are a crucial resource within the institution, bringing to bear their knowledge, skills, expertise, and connections for the overall development of students, colleagues, institution, and the wider community. Professors contribute to research, teaching, and administration, and mentor colleagues in the development of core competencies and skill sets. They develop the necessary synergies and connections to attract large research grants from outside the university and engage in policy development. Professor Ramsubhag epitomizes these attributes in his teaching, research, and publications. Further, his research on microbiology speaks directly to several of the UN Sustainable Goals, including Goal 2, Zero Hunger, Goal 3, Health and Well-being, Goal 6, Clear Water and Sanitation, Goal 13, Climate Action. The University of the West Indies has a major role in addressing issues affecting country, region, and the world. Besides our core remit of teaching and preparing our students to take on major roles, an enormous responsibility of the St. Augustine Campus is to undertake research, disseminate findings, and engage stakeholders and the public. Well, the Open Lecture Series is one of the many ways in which the St. Augustine Campus directly addresses this mandate. The Open Lecture Committee falls under the auspices of the Campus Principal's Office, and I'm therefore delighted to invite to the podium our very active and inspiring Pro-Vice-Chancellor and Campus Principal, Professor Rosemary Bell Antoine, to bring opening remarks.
Thank you, Master of Ceremonies. Good evening, ladies and gentlemen, colleagues, members of the media. And I'm only going to say one special good evening, and that's to you, Professor Ramsubhag. And on behalf of the St. Augustine Campus of the University of the West Indies, I'm really very pleased to welcome you all to this inaugural Distinguished Professorial Lecture by Professor Adesh Ramsubhag. And we are here this evening to speak on an area that many may find obscure, a microscopic world that affects our very lives. He's a specialist in microbiology and biotechnology, and he's fascinated by the 1 trillion estimated species and 5 trillion cells, microorganisms that dominate our world and impact our lives. How am I sounding, Dean Coburn? As though I know what I'm talking about? Actually, I don't. And this is as far as I'm willing to go with regard to these organisms, since I'm very much aware that my area is law. So I am here to listen to the unquestioned expert in this area. Professor Ramsubhag's profile speaks to a rural upbringing in a large family. Well, I have that in common. And among a community whose mainstay was agriculture. In such an environment, an individual becomes very close to nature and interested in the nature of things. And I think many of us can relate to that. He was able to clearly see that hard work often brought reward, but sometimes it brought heartbreak when nature did not cooperate with the plan. A degree in agriculture seemed the most obvious course, and his life trajectory seemed clear. That is, until he came to the St. Augustine Campus. UWI has a way of doing that to questing individuals, always surprises in the life of academia. He speaks of mentors, like our very own distinguished and impactful scientists, Prof. Pathmanathan Umaran, you know him from the cocoa research and hot pepper and all of these things, and the late Dr. Alfred Donower. I don't think you can get any better mentors, Prof. And that also struck a chord with me since we just launched a St. Augustine mentorship program last week. Mentors volunteered to encourage, share their experience and knowledge, push as required, and be listening sounding boards. Very important to an academic community. I have no doubt that this was Professor Ramsubhag's experience on his way to where we are now at his inaugural professorial lecture, and where he has now become the mentor to others. This evening is a personal and professional landmark moment for Professor Ramsubhag. This inaugural Distinguished Professorial Lecture allows him to share his achievements in research, engagement, teaching, as well as his practical experiences in his study of this all-important area. His family, friends, colleagues, and the students are present this evening to share in his achievements. For some, it will be an opportunity to get a behind-the-scenes glimpse of an academic's life and the application of his area of study to our everyday lives. This evening, I hope you will also understand why increased funding is so important if we are to advance scientific research and innovation, so that one day this country and this region will enjoy economic and ecological self-reliance and sustainability. My congratulations, Professor Ramsubhag, and my best wishes for the future. We look forward to your continued excellence, and more importantly, from learning from you, beginning with this evening. Thank you.
Professor Ramsubhag is a professor in the Department of Life Sciences in the Faculty of Science and Technology at the St. Augustine Campus. I'm pleased, therefore, to invite the Dean of the Faculty of Science and Technology, Dr. Brian Coburn, to introduce our featured speaker, Professor Adesh Ramsubhag. Dr. Coburn, as Professor Ramsubhag, I must say a few things, obtained his PhD at the St. Augustine campus of the University of the West Indies. Also, Professor Ramsubhag also obtained his PhD at this university. His interests includes studies in metabolic diseases, and he has published widely. He has also served as campus public orator, and as I was telling Dr. Coburn, there's a saying that when he speaks, he sings. It is my pleasure to welcome Dr. Coburn to the podium to introduce our professorial inaugural speaker.
Thank you, Chair, Professor Chady, Pro-Vice Chancellor and Campus Principal, Professor Rosemary Bell-Antoine, our Deputy Principal, Professor Indar Ramnarayan, our featured speaker, Professor Adesh Ramsubhag, Professor of Microbiology and Biotechnology, other faculty deans, heads of departments, members of the private and public sector, family and friends of Professor Ramsubhag, specially invited guests, members of the media. Good evening. It's a little known fact that I was Professor Ramsubhag's office neighbor for many years. So I have to start by getting some things out of the way. So, those of you who think Dr. Ramsubhag is a meek, humble guy, obviously don't know him very well. You also probably don't know any farmers. Oh, Adesh wears a good cover, but anyone from a family of 9 siblings and practicing farmers must truly be made from the toughest stuff on earth. Resilience is a superpower after all, and we're in the presence of someone super. And there's a contradiction or two as well. He tells us that he never liked the rigor and rote structure of school. But if you see him in a tennis ball, or as we say, wind ball cricket fet match, bowling the ball to great effect, and by that I mean getting people out, while everyone else is pelting, you'll understand that he has the stomach for rigor and meticulous action. That's also on display in his love of classical Indian music and his accomplishments on mandolin and guitar. He sings too. This is someone with a genuine love of life that manifests itself in multiple ways. Fortunately for us, one of those ways is in a love of science, of discovery unbound, a passionate spirit more at home in the lab than in any office. Well, unless that office is populated by students and colleagues discussing the work. As he describes it, his family always appreciated knowledge, learning, and ideas. We are in their debt. Professor Adesh Ramsubhag's CV reports that he is the author of 79 peer-reviewed journal articles, 2 rough-read book chapters, 14 conference abstracts, published in peer-reviewed journals and proceedings, 43 refereed conference presentations. He has successfully supervised or co-supervised 11 PhD candidates, 24 MSc research projects, and numerous undergraduate research projects, some of which matured into greater things. He has contributed to the establishment of microbiology as a discipline in its own right. And moreover, he continues to ignite the passion for microbiology among the student body, nurturing and inspiring the next generation of microbiologists. Professor Ramsubhag lists his research interests and experience as environmental microbiology and environmental health. This is enough to consume the time of several academics, but it is only the beginning. Novel antimicrobial compounds and bioproducts, bioremediation, bioaerosols impacting on indoor air quality, antimicrobial resistance, vector-borne viruses, crop diseases and health, molecular characterization of pathogens of tropical crops, biological control of pathogens, plant biostimulants. No, the list is not exhaustive, but you get the picture. This is a Renaissance man. He has broad interests that he entertains deeply. He is also active in popularizing science on such topics as microbial biodiversity, the commercial seaweed industry, and pesticide use in crops. He has also served the University of the West Indies faithfully as head of the Department of Life Sciences from 2015 to 2018, and later as Deputy Dean for Graduate Studies and Research in the Faculty of Science and Technology. That was actually earlier, from 2012 to 2015. Now, if all this strikes you as evidence of excellence, you're in good company. His body of work was recognized with an Anthony N. Sabga Award for Caribbean Excellence in Science and Technology in 2018. And in 2019, he received the Vice Chancellor's Award for Excellence in Research. This includes his team's identification and chemical modification of biomolecules from bacteria, which can serve as adjuvants to existing antibiotics. Ladies and gentlemen, from humble beginnings to the lofty heights of scholarly accomplishments, Professor Adesh Ramsubhag reminds us that it's not always how you start that matters, but how you finish. And just to be clear, Professor Ramsubhag is not finished. He's about to shift into a higher gear. Thank you.
Thank you, Dean, for those kind words. A little bit exaggerated, but nevertheless, still appreciated. Okay. Pro Vice Chancellor and Campus Principal, Professor Rosemary Bell Antoine. I was told Antoine. Deputy Campus Principal, Professor Inda Ramnarayan. Dean of Faculty of Science and Technology, Professor Brian Coburn. Colleagues, students, friends, family, ladies and gentlemen. Pleasant good evening, and thank you very much for your presence today. It's really, really appreciated. So, as you will see, my title today is Journey Through the Local Microbial Landscape, Challenges and Biotechnological Potential for the Caribbean. I really tried to get away from this presentation. So when Prof. JD contacted me in a weak moment, sometime back, I said, okay, well, maybe. So then time kept coming, and I said, you know what excuse can I give? And then before I can tell him anything, he said, campus principal is coming to your presentation. Well, that blocked me, that stopped me. I say, well, it would be too disrespectful for me to cancel at that time. But nevertheless, I really appreciated the opportunity to share my experiences. Oops. Right, so this is basically the outline I'll be talking about. I will talk a little bit about my journey into academics, accidental as it may be. I will give a little background, not too much. I'll try to be as simple as possible, but talk a little bit about the microbial world. And I will highlight a little bit of unique features that we observed in the local Caribbean landscape. And then I'll give a little summary of my research at UWI, highlighting certain aspects. And I tried to include all my students, but it was not possible because I highlighted little bits and pieces here and there. And then a little bit about the challenges that we face in moving research and technology and innovation forward in this region where we are starved for funding. Okay, so again, you would have heard I came from a very rural background, very simple village. And I know many of you all might be shocked, as one of my students recently. So I told her I went to school bare feet. And people can't imagine that, but it was in my era. A little bit late, but we were very country, you know, very rural. But we lived simple lives, and we were very, very happy. You know, villages like ours, you know, academic studies and so on wasn't a priority. Everybody was expected to get into farming eventually. And I had a special aversion for school. I really hated school. And I think I had a traumatic experience. You know, when I try to reflect back, it's a little bit hazy. But I remember, like in year 2 or year 3, I got promoted. I came out top in class, and they put me in another class. And I came out last in tests next time. And it's not easy to, in a countryside, you know, villages. Everybody knows everybody's business. And, you know, and I suspected that was the trigger for me to not like school. But I labored on. You know, I was in a family. My mom believed strongly in education. So they made all the efforts so that I will go to school. And I labored on. I eventually went to secondary school. I finished high school. I ended up in A-level class in modern studies of all matter. I wasn't even a science student. But nevertheless, you know, I persevered as much as possible, you know, as much as I was able to go to school. But I love staying home. I will make excuse. I will feign illness sometimes. You know, I'm not well. And when I stayed home, obviously, you know, we are agricultural village. So naturally, you know, there will be activities, and I always find myself in the field. And I was happy in that environment. So and the community itself had a very, very, very big impact. While I hated school so much, I love community life. You know, like in the evenings, we play cricket, football, we go fishing. At that time, I wasn't a vegetarian. Go fishing, play music. That was our activity. And then in the night, we used to be, you know, having a lot of cultural activities and so on. So that was good life. But nevertheless, I ended up in academic environment. So after A-levels, my performance, not surprisingly, wasn't that good. I couldn't get into a good program in university. So obviously, the next thing came, and the obvious thing was to get into farming. So I was a full-time farmer for 2 years at least. And those crops there that I showed, it was deliberate. That was my existence. Sugarcane, I love sugarcane operation. Then, we planted bodi, cultivated rice. I love planting rice in the mud. I never liked harvesting the rice though. But planting and playing in the water and the mud, that was really fun. And then watermelon, that was our main crop as well. And it was all well and good. And then eventually some relatives came and they said, why don't you go and do your BSc in agriculture? Well, I said, well, I didn't do sciences. So they said, well, no problem, you can go through the N1 program, the pre-science. I said, okay, yes. No problem, I want to improve my farming skills, obviously. So then I decided to sign up, and obviously it was difficult at that time in the family, but they made the sacrifices to send me. And I was introduced to the sciences. I did the N1 program. I went into the BSc program, and then I became fascinated. I became overwhelmed with the sciences. So I found my calling. Maybe the environment was also important. It was more conducive environments. I ended up with a close group of friends who kept pulling me. I didn't have a choice. They kept coming, like, if there's work to do, we'll all do it together, right? So I started to enjoy, and then I was fascinated with lecturers. Obviously, my Professor Umaharan was one of those big inspirations. Professor Dr. Donowa. Those were my areas that I liked. But surprisingly, one of my favorite lectures and one that I enjoyed a lot was Morgan Joe. He was out of the box thinking. He could be critical, and I appreciated it. I really, really enjoyed those vast and different types of courses that we did in the agriculture program at that time. It was very, very science-based, and it really fit into my likeness a lot. So I have full appreciation for the university and for my lecturers. In our tradition, we treat our teachers like God, and I consider my supervisors and my teachers as God. So anyway, as I said, I ended up now fascinated with sciences, and especially microbiology, genetics, biotechnology. Those are my interests. I wanted to understand the core of biology. And why I went into this area, I wanted to do genetic engineering. I wanted to be able to play with the DNA and manipulate and create new forms of life and so on. So that was my interest, actually. So I started a master's program, and then I went into the PhD. Again, I enjoy every bit of it. And importantly, and with the support of my supervisors, I was able to use most current modern molecular biology skills, or develop those skills and apply those skills to my research. And I never regretted it. It allowed me to keep up to date with technology, to have a passion to keep up to date and apply these new skills to solve societal problems. After I finished, I worked at Kerry for 3 years. And that itself was a marvelous experience, because it allowed me to interact with companies, with individuals, to solve problems. And that was my strength, actually. I don't like writing too much. Reading is sometimes a challenge, but my playing field is give me problems to solve. That is my nature, actually. But I got the opportunity to come back in campus, and I was missing applying these new biotechnological tools that I didn't have access to then. So I came back to campus and joined the department as a lecturer in microbiology. So now I'll go a little bit into the technical aspects of the presentation about the microbial world. And we truly live in a microbial world. Our existence as humans is just is incidental. It's of no significance when we compare to the biological world. And microbes obviously are organisms that we cannot see, but they are all around us: bacteria, fungi, viruses. Each breath I'm taking, I'm shedding possibly dozens, hundreds of microbial cells. And I'm also taking it. So we are all sharing our microorganisms in this room, believe it or not. Yeah, so we have something connecting us all in this room right now. All right. And given the fact that microbes have been around for 4 billion years, almost 4 billion years. In fact, some people think that it is even before, earlier than 4 billion years. So they have had so long time to evolve. And from that evolution, we now have so much diversity, mind-boggling diversity. And as it's mentioned here, we have, right. Theoretically, we have over 1 trillion bacterial species present on Earth. And that's amazing. All right? Every day, and you'll see from some of my research as well, every day we discover all of these new organisms. And get it, 5 million trillion cells. You were off by 1 million trillion campus principle. 5 million trillion, 10 to the power 30, and that's mind-boggling. All right? If you think about the amount of humans we have on Earth, maybe 6.5 billion thereabout. But if I go to the lawn outside and take a little pinch, a little speck, we will have more microbial cells in that little sample than we have human beings on Earth. Okay? Because every gram, every little small quantity, every gram of soil, we can have 10 billion cells and up to 50,000 species. And that's an average set of data. In our tropical environment, it could be 10 times as many, all right? So it is amazing. And to put things in perspective, let's look at the human being. Human beings, right? Our gut contains, on average, 10 to 100 trillion microbial cells. That is 10 times as many human cells that we have. Okay? 1,000 species and 3 million microbial genes. And that is, again, 150 times as many human genes that we have. So we are microbial more than human. We are supraorganisms, as the term is referred to now. We are humans, yes. But we are beyond humans. We are humans plus so many other things. And these organisms significantly impact our health and well-being. So, again, when people think about microbes, they think about germs. They think all microbes are bad. But on the contrary, most microbes are beneficial to us. And only a tiny, tiny, tiny little fraction of these organisms can cause disease. In fact, our existence depends on the microbes. In fact, based on biological science, right, we would have evolved from these microorganisms. So the food we had for lunch was digested using enzymes that were present in microbes 3 billion years or more. Okay, that's our connection with the microorganisms. All right? So they do have many, many benefits to us. I mentioned about the gut microbiome, up to 100 trillion cells. So these organisms, humans and all other organisms, would have evolved or co-evolved with these microbes. So we cannot separate ourselves with the microbes. And we depend on certain microbial composition to maintain our good health. So for instance, if we eat a bad diet, our microbial, or what we call the microbiome, the complete community of microbes, they go into imbalance. And that has been linked to a lot of diseases, dozens of diseases. Diseases that are linked to the gut, like irritable bowel syndrome, for instance. Obesity is linked to the microbiome. Cancer, obviously. Neurological diseases, diabetes, a whole long list of diseases are now linked to the microbiome. And the microbiome is influenced by the food that we eat and also the environment. So that link is clear. It's beyond theoretical now. We have strong evidence for some of those diseases. even the skin microbiome protects us. And there's a community of people now, a growing community, that don't bathe. Believe it or not. They don't bathe because they think using soaps and detergents and so on will alter the microbiome and affect their health and the quality of their skin and so on. So sometimes if I'm doing work and my wife is reminding me time to go and bathe, what I'll do? I'm going to join that community. All right. So this science now is driving new technologies even. Like the gut microbiome type of studies has led to development of fecal transfer technology. People with certain diseases like irritable bowel syndrome and even certain types of infections, they take fecal matter from healthy individuals and transfer it into these patients with very, very good amount of success. Okay, there are other benefits, but I won't talk too much. I have a lot to talk about. So just to go quickly, they protect plants against diseases. They are critical to soil fertility and plant nutrition. Without them, we would not have any plants. Degradation of harmful substances, toxic substances. If the microbes weren't there to degrade organic matter, we would have been living on miles of layers of plant and animal debris. That is how important the microbes are. And then obviously they have source of food. We eat mushrooms. We use microbes to bake bread, to brew beverages, to make alcohol. Some of you might like that. I'm looking at my nephew. Say thanks to the microbes, right? And then obviously a source of drugs. 2/3 of all my antibiotics have their origin in microorganisms, right? And industrial products and so on. These are very, very critical organisms. So in terms of the microbial, the unique features of the Caribbean microbial biodiversity, we know that the Caribbean region is a biodiversity hotspot. But this is usually based on higher organisms. We know we have so many species of plants and birds and insects and so on, animals. It's enormous. But it is obviously nothing compared to the microbial. If we plow deeper into the microbial biodiversity, as we have been doing, it's mind-boggling. And I don't have graphs to show, but I could just mention that we did a small number of samples in Trinidad. We explored the diversity of microbes in different types of ecosystems. But in few ecosystems that we looked at, it is enormous. It's like we're looking at, I mentioned up to 50,000, 40,000 species in some samples. We were getting hundreds of thousands of species in a single sample. Okay? So it was very, very enormous. And my colleague at that time I was working with from abroad, they had now completed a study where they traveled around the globe, collected marine samples in a yacht. I think they went actually twice. And then when he looked at my data, and in fact, I was very early in my learning bioinformatics skills at that time. So he also helped analyze the data. And when he looked at the data we got and what they got from their trips around the globe, he said that there is no comparison with the amount of diversity we got compared to the very little they got in that voyage around the world. It is that enormous, just a handful of samples. So what does that mean? We have tremendous potential. So imagine a single gram of soil has 10 billion cells. A gram is like, small little bit, right? And has 100,000 species. Imagine the type of chemical warfare is taking place. Here, somebody lives by the neighbor, 100 feet away, they're fighting. All right? Imagine in one tiny little gram, you have 100,000 different types. And so many billions of cells. So imagine the type of warfare you'll have going on there. So that chemical warfare now that is occurring in this type of environment is a driver for novel antibiotics, novel bioactive compounds. So hence, there is tremendous opportunities to exploit this biodiversity, these bioresources, for discovering new drug molecules, for instance. That's just one of the applications. There are so many other biotechnical applications that I could talk about. But obviously, when the organisms are fighting against each other, one will produce an antibiotic, and the other will produce a chemical to counter the antibiotic. So what does that mean? You get new antibiotics, but you get new forms of antibiotic resistance. So these antibiotic resistance now come into the clinical isolates, and they cause more serious disease. All right? So that's the trade-off. And my research now has been looking at both aspects, the good and the bad, and the ugly as well, right? So basically, I can divide my work into two different categories, broad categories: detection and characterization of harmful organisms that may cause disease, for instance, to humans, to animals, to plants. I've been a general microbiologist and working with people with different interests. I was able to, get involved in so many different types of research projects. And it's also like a payback. I am appreciative of the influences my supervisors and my lecturers would have had, and I benefited. So it means that I have to return the favor. And that has been a driving force for me. I really love working with students, and I'm highly motivated with students and their research, and I get the most happiness when I see the students evolve and grow. As you will see here, most of the research is collaborative, and I've worked with a fair number of students, and working with different organisms, bacteria, fungi, viruses. And saying fungi, my daughter gave me a joke. So why does a mushroom get invited to the party? You all should have known that. Right, he's a fun guy, right? Yes, very good. Right. Okay, right. Again, and the other arm that I look at is the exploring biotechnological applications, right? How can we generate beneficial products or processes and utilize these to solve problems in society? And for that, we have three different sub areas: bioremediation, plant biostimulants to enhance growth and protect plants from diseases, and then our main area, one of the main areas, is antimicrobial drug discovery. And we use a range of different techniques, and I won't go too much into that, but some of the techniques we use involve culturing and growing organisms. That is why microbiologists are well-cultured people. So we culture these organisms, but we also use a lot of molecular-based techniques where we analyze DNA and RNA, the nucleic acids, the genetic codes of these organisms. And in fact, I spoke about that 100 million and billions of cells, but most of them are not culturable. So if we really have to understand and exploit these organisms, we have to go at the genetic, the DNA level. Okay? All right, so now I'm going to talk about little sub-areas. So here is one of the areas we worked on, environmental monitoring. And as I said, it was difficult for me to select what aspect of the research to present because I wanted to be fair to all my students. So I listed the different students working in the different categories, right? So these are mainly MPhil and a lot of MSc students working on different aspects of monitoring, biodiversity assessment, pollution monitoring, in freshwater, marine water, soil, and air. And obviously, I believe that the sum of the whole is bigger than the individual parts. Teamwork is the way to go, especially in a resource-limited environment like us. How can we get access to equipment? How can we get some little bit of resource somebody will have? Is we have to collaborate. We have to share. So a large number of collaborators from campus, from UE, from outside our department as well, and in fact, many from external universities as well. So this is just one little bit of data that we have, one study actually. We cannot go too much. We have been monitoring water quality in water bodies, fresh water, marine water, as I mentioned, rivers and ponds. And this is a typical type of result we will get. Our rivers and our bodies of water are heavily polluted. We have a lot of effluents coming, raw sewage will be getting into the water, and it is destroying the environment. And in many of these sites, you have effluent entering the water, and 200-300 yards, what they are doing, they are taking water and using it for irrigating the crops. And then you buy your fresh lettuce, and you just shake it under the water, under the tap, and you eat it, right? That wouldn't remove all the microbes. And especially this one is a frightening one. Here we looked at just pollution level using indicator bacteria. But here we actually use this DNA-based methods to detect the pathogens that are present. So these are potential pathogens because we just analyzed the DNA. We didn't isolate the cells to see if they're actually pathogenic or not, right? But yet, nevertheless, we have seen some frightening type of data where a lot of different pathogens are present, and they are present obviously in the sites that have higher pollution, all right, which is not surprising. So this is a typical scenario in our freshwater, even in our beaches and marine environments. Similar type of data we have been getting. But we use deep metagenomic sequencing where we looked at antibiotic resistance as well. And these, in about 5 or 6 samples, we got over 300 antibiotic-resistant genes. All right? And most were found in 2 heavily polluted sites. One site had raw sewage entering, and the next site had effluents from an industrial meat plant. All right? So not surprising, again, antibiotic resistance getting. And again, most of the resistant genes were for commonly used antibiotics. Some are even 3rd and 4th generation antibiotics that, really, they are very important for preserving health. And again, think about it. This going into the vegetable, green vegetables, and when you eat it, you get not only pathogens, but antibiotic-resistant pathogens. If you get sick, you have difficulty in treating these infections. Okay, so another one on plant health. Again, I'm just giving 1 or 2 bits in each subcategory. So we look at broadly in different areas, disease prevalence and management, copper resistance as well. And again, a number of students, many PhD students in this category, several MSc students. I've seen some here today as well that I haven't seen for a while. All right. And again, a broad range of collaborators from within UE, as well as some foreign collaborators. And again, I'm just going to report on one study. We have been monitoring copper resistance. Just as we have antibiotic resistance in human and animal pathogens, we have pesticide resistance or fungicide resistance in pathogens that affect plants. So the same trends we are seeing. So copper-based fungicides are used as important agents to protect plants against diseases. And resistance have been building. And we have seen this. So in 2000s, we did a study, and 63% of the isolates were copper resistant. Now, recently, my student is doing a similar project. 100% we have been finding in many fields. Okay? And then what that will lead to? Farmers use copper pesticides, it doesn't work. Then they use more. Higher frequency and higher dosage. And they use cocktails. And all of those have very, very serious impacts and implications for health as well as human health and environmental health. Okay, again, this is something we need to look at. And again, we went deeper into the genomics. What are the genes that are present? And we are finding a lot of unique genes. And again, if we can remember earlier on the mind-boggling diversity we have in this region, we also get in unique forms of pathogens. Maybe we don't know, they might be difficult to manage, so this is something we need to go deeper on. What sort of challenges that these new pathogens are bringing forth? All right, we may need new strategies to manage these. So here's an example. So this is a gene, a copper-resistant gene cluster. We see this is a phylogenetic tree. So all the known genes fall in this branch. So this tightly packed branch, which means that they are closely related. But look at ours. Ours keep completely out, which means it's a divergent gene. Again, we don't know the impact of that. We have to investigate. These two here are not even the same gene. They are related genes. All right? So it's so diverse, it's completely separated from the common genes. And not only that, this is copper-resistant genes. We also see a lot of unique genes that affect pathogenicity as well. All right? So again, these are things that we need to investigate. So we have also been looking at integrated disease management, and this is a project I work closely with Prof. Jaya. So farmers need alternate strategies and support to treat and to manage diseases. So here, what we did, we selected environmentally friendly alternatives, more biological, minimal use of inorganic fertilizers, which are harmful types, inorganic pesticides, right? And we have different combinations that we code them T1 to T8. T9 is the control, right? Things like using manure, biological control agents. Here, these are microorganisms, fungus, and bacteria that we isolated and test and confirm that they are able to control diseases. So we use that in the mix as well. Again, more opportunities for local microbial resources where we could convert to products. So here we have examples of biological control agents. And again, a range of different, more environmentally friendly options. We treated those plants like, tomato fields. And this is the results. Here, each bar here is a different treatment combination, right? Environmentally friendly treatment combination. And you can see here, the bars here, the axis here represents the amount of disease, basically. You can see here, this bar here, this combination number 1, I think, almost eliminated this disease here, early leaf blight. And then similar results we saw, reduced disease with different treatment options. So again, and look at it, it's not here, but these treatments were given like every 2 weeks, all right? Compared to sometimes farmers now need to apply every week, more than once per week, sometimes they apply these fertilizers. Again, if we move more towards these biological control or environmentally friendly options, we have a better option of controlling these diseases, and in a way that will be safer for humans to consume and the environment as well. So another area, broad area, is human and animal health. And again, we looked at different types of projects, again, detecting and characterizing pathogens, animal microbe interactions, antibiotic resistance, use of probiotics. We did develop some probiotics that give protection against diseases, and biological control as well. Again, a number of PhD students, some MSc students, and collaborators within campus in different departments, and also externally as well. So this, again, is just one slide to present some results on viruses. I know we are now coming out of the pandemic, and everybody is so sensitive and aware of viruses. So these are two viral hunting projects. Here, one, we looked at virome studies on mosquitoes, Haemococcus mosquitoes. This is a mosquito that transmits a disease called myrovirus. So we were looking at the prevalence of the mosquitoes as well as what viruses that they may have. And again, a few samples, about 10 samples, less than 10 here. We got so many different types of viruses, many of human health concerns as well. All right? But importantly, what we found, previously these mosquitoes were limited to certain areas in Trinidad, like Northwest Peninsula, but now they are all over, all right? And importantly, we found that the mosquitoes are feeding on birds and rodents, whereas previously they were thought to be feeding only on monkeys. All right? So it means that if there's a disease epidemic with myrovirus, they also transmit the yellow fever. All right? Or if that virus jumps a host and goes into birds, and birds are all over, and the mosquitoes is all over, it means that we become more susceptible. All right? So the ministry is aware of this, and obviously this will be taken into consideration in their strategies for disease management. One more, we have a certain amount of people that are affected by paralysis, polio-like symptoms every year. So Suman here has been working on this project where we have been looking at viruses that are related to polio, all right, and try to establish whether there's a link in that, whether or not the viruses are able to cause the symptoms. That part of it isn't complete, but we have found a strong association between, or let's say strong occurrence of paralysis with many of these different types of viruses. And importantly, we found many new strains, okay? So that is a project in collaboration with CDC and CAFA-based project as well. So that's just one aspect of the research on human health. We also work with food and water quality, and again, we collaborate with different faculties, a number of students, a number of PhD students here, and collaborators from within and outside of the campus. So this is just some data on a recent study conducted on water quality and the quality of domestic water in rural communities in the Caribbean. So we looked at different types of water: rain harvested water, water from tank, water from rivers and so on that people collect and use, and even some treated water. And we are seeing fair numbers of these samples have contamination, right? Here shows E. coli, which is an indicator of fecal contamination, plus they can also be pathogenic. But we also applied deep sequencing-based technologies. And again, we are seeing signals of many pathogens. We don't know if they will actually cause disease, but the signals are there, right? Which means these need further investigation. All right. So this one here is one I'm also excited about. This is the last segment, talking about biotechnological applications. The previous section, we talked about disease-causing organisms, harmful organisms, how we can detect them and manage them. Here is where we try to develop solutions for problems. So again, a number of students, PhD, MPhil students, MSc students have been working. Different collaborators from within campus, different departments. So this is one area, bioremediation of petroleum contaminated soils and environments. So we have been collecting these organisms from these polluted sites. So these organisms have the ability to degrade these contaminants, harmful substances. And we develop consortium, combinations of bacteria that we now test to see how they can degrade the oil in contaminated soil. And again, you can see, this is the control here. And some consortia here we have, this is a fungal consortia, almost 90% degradation within 28 days. Okay, this is rapid. This one is bacteria, 21 days, and again, about 85%. Which means these are powerful technologies, potential technologies that can be applied for treating these contaminated environments. Next one is development of these biostimulants and bioelicitors, and this is a project that we work with Prof. Jaya. So again, we know about the sargassum problem that we have. So for a long time, a long, very, very long time, seaweeds have been used as plant supplements, right? As manure, as compost, and as biostimulants. So this is a project that Prof. Jaya had started that I became a partner with more than 10 years ago, even before sargassum became a problem. And obviously, once sargassum became a problem, we decided, okay, let's see how we can develop this sargassum into a biostimulant product for crop, for enhancing crop growth. So we did develop, we used certain solvents and so on to extract these beneficial compounds. We developed a commercial product. This should be commercialized pretty soon, hopefully. But we are working towards it. So this is a product here. You have a liquid formulation that can be applied to leaves of the soil. This is a label. Oops. Oh, right. So this is just some of the results. I don't know if you can see well, but this is a control here. This tiny little plant here. This is a sieve, I think. And this is our product. You can see the difference. This one is celery again. This is the control, and this is applied to the product once every 2 weeks. A small amount, 5 mils in a liter. Very, very small amounts. This here is lettuce. This is control. This is a commercial product in the market. And this is our product. All right? So it's fascinating. We have, I believe we have some farmers in this room who have been testing it. So they have received this product and they have been reporting to us. And so far, the results have been very, very, very promising. Okay, so the last one I want to talk about is antibiotic resistance and development of new antimicrobial drugs. So we know antibiotic resistance is a real problem. Currently, around 700,000 people die every year because we don't have effective antibiotics to treat these infections. And it is projected by year 2050, we'll have 10 million people dying every year. Because the problem is getting worse. Antibiotic resistance is spreading rapidly. And you could just look at the problem we saw with the pollution from the sewage and so on getting into the water. So it's spreading like wildfire. So one of the solutions is to find new antibiotics or new forms of treating antibiotic resistance. So this is one of the projects that we would have started long time. In fact, my first project when I joined as a staff was on antimicrobial drug discovery, working with my mentor, Prof. Maxwell from chemistry. So we developed this close relationship since that time. So I just am presenting here two results of two studies, 2 PhD students. Trisha Dobson from chemistry, she was resident in chemistry, working on isolating these active compounds from a bacteria we isolated from the pitch lake. All right? So in that, she got 22 active compounds, and then 7 were novel compounds, or new compounds. All right, but most of the discoveries were towards the end of the cycle of the program. So we didn't have time to do further work. We just discovered these organisms, and that was end of the project. If we had funding, then we could have continued, right? So our research is restricted by dependence on students, because there's no funding to have any independent research apart from what we can get with students. The other project by Antonio Ramkisoon, again, he's finished and he's at Harvard right now. He again isolated several new compounds, one of which was a new antibiotic of a known class. But importantly, we found 2 new classes that can be considered as adjuvants to antibiotics, which means that they themselves do not have any antimicrobial activity. The bacteria grow happily in the presence of these organisms. But if we add them to antibiotics, then that makes the antibiotic more effective. And even if the bacteria is resistant, what it does, the adjuvant knocks out the defense capability of the bacteria. And then the antibiotic will work happily. So imagine like you have an army. Army has the most modern equipment, and they are asleep. Somebody can move and kill them easily, right? With inferior weapons. So this is what happens here in our case. We have these strong antibiotic-resistant bacteria that can kill someone, but once we add our agents, our adjuvant, what it does, it knocks out the recognition system of the bacteria. The bacteria doesn't know, okay, that harmful substances is coming to kill them. So they are not able to defend themselves, so the antibiotic works. Even if if the bacteria is resistant to the antibiotic will work, all right? So this is some data, and the difference between this and the previous project, the discovery was made early on, and a lot of the research was on the developmental side. How can we convert this now, move it closer to our product? So here we have different compounds, and these are MICs of antibiotic, minimal inhibitory concentration. So it means here the bacteria needs up to 128 micrograms per mil for it to be killed. When we add a little bit of our adjuvant compounds, the MIC of the antibiotic reduces. And this was preliminary screening. This wasn't optimized data as yet. So we see, for instance, it moved from 128 by itself. When we add our compound, it moved to 4, well within the range for using as a therapeutic agent. Some ways even 120 it goes to 1. Some even after optimization it went much slower than that. And different antibiotics. This is MRSA, methicillin resistant Staphylococcus aureus, vancomycin resistant Enterococcus. And again, same thing happened with few tests that we did. From 64 it went to 4, from 64 it went to 1.5 and so on. So again, this is very interesting and very fascinating to us. So we looked deeper into it as well. So we did a lot of different studies on safety and efficacy. So this is one of the mice trial we did. We inoculated mice with a heavy dose of the pathogen, a lethal dose. And then we gave different combinations, one with antibiotics alone, one with antibiotics and our compound, and then one with the adjuvant alone, not with the antibiotic. So here, this is the result with the antibiotic alone. Lethal infectious dose plus ampicillin, which is a common antibiotic. And look what happened. After 1 day, all mice died. All right? Here is with our antibiotic plus one of the adjuvant compounds. See, all survived, and they were very happy. if I could say so, right? They were very active and vibrant and moving around, This here is when we put the adjuvant by itself. It did give a little protection, but some survived, but they weren't too well. They had to be euthanized immediately. Yeah, they weren't happy. All right. So, right, and we did other studies. We validated or determined what was the mechanism, and this is how we determined, okay, it was knocking out that signal component, that ability of the bacteria to detect signals. Okay, there's stress, there's harm, let's arm. Okay, let's trigger these defenses so that they can counteract the antibiotics. And different independent studies, we look at transcriptomics, look at how genes are expressed. We look at in silico molecular, in silico docking computational models. We did protein binding assays as well, and all point to the same mechanism. All right, and again we did safety and toxicity profiling. So they have excellent safety profiles. They were all eating well and gaining weight as normal, and no cytotoxicity in human cells. And no hemotoxicity as well. So again, it's a good candidate to move forward, but still it is early in the development process, and it will take significant amount of investments to take it into the pharmacies. Okay, and that brings me to the last slide on this presentation. Well, last technical slide. So how can we move research and innovation in the Caribbean? So I have a colleague who was doing a similar research in the US. And once, we met and we were talking. And when he found out our results, he was so amazed. He was in a startup that they got 85 million US. And he said they didn't even reach close to what we reached. And they had spent 50 million at that time. All right? And we took our research, and a lot of the development here, we achieved with zero funding. Zero funding. We got some funds from UE afterwards to continue, but the initial discoveries was just based on motivation, hard work, sleepless nights. And this is what it takes in an environment. How can we compensate and do research in an environment that is not conducive to research? All right? This is where we need to come out of the box. All right? This is where teamwork is also important. So for instance, for that project, we use old chemicals. We purified the chemicals, expired chemicals. We used expired consumables that were being given away by other labs. Okay, and thanks, CAFA. We got some resources because they're in a regulatory lab, and after it expires, they cannot use it, right, for legal purposes. But we are happy to take it. And being a microbiologist, when I was in Curry, one of my functions was to establish shelf life. All right, so we could do the quality controls and know if it is okay to use. And in fact, for research, it's not critical. I want to synthesize a compound. If I get old compounds that is degraded, it's okay, I'll purify it and I'll continue my synthesis. All right? So this is how we were able to achieve some of these objectives. So again, coming back to this region, there's huge potential, and there's also possibility of biopiracy. People do come. I had a friend who was offered a job in the US to come and bioprospect in the Caribbean. What it means to come and collect biological material and take it back to the lab for this research to discover new drugs and so on. All right? Which means that we have to find somehow, I don't know if it is possible, if it is a dream that may not be fulfilled, how can we get funding to carry out some of this research for discovery as well as development? And that is a major impediment. All right? And as with the first cycle with Tricia, it stopped. Potential is there, but we cannot continue. And then that is just two organisms that the two students worked in. We used to be isolating hundreds of bioactive or antimicrobial microbes producing antimicrobial compounds. So there's going in a fridge, or they may be discarded because we can't keep forever. So all of these are potential resources that are not being utilized, capitalized. And just to mention, Trinidad and also Jamaica, our funding for research and innovation is very, very tiny. It's 0.06% of GDP, which is, in terms of, I'm not an economist, I don't want to give wrong figures, but it's probably less than 20 million US. All right? Compared to developed countries, there's a strong relationship between investments in research and innovation and development and efficiencies. Okay? Compared to Singapore, 2.2%, but they have the highest per capita investment in research and innovation. But that 2.2%, comes out to 10 billion on a yearly basis, 10 billion US. So equivalent, if we consider our smaller size to Singapore, 5.5 million or so, the equivalent amount we have to spend to match Singapore is quarter of our GDP. All right, so that's the big difference. And look at the big economies: 3.5% in US, 513 billion. South Korea, 4.8. This is one of the highest percent GDP in research, up to 100 billion. So there's a reason why these countries obviously are first world, and they are progressive, because they invest a lot in research and innovation. Okay, and we need to probably take a leaf of their book if it is possible, right? Again, I don't know if it's a pie in the sky type of dream, but we could only dream. Again, significant investments are needed to take products to the market. But the potential benefits are tremendous. So for instance, if you can get one drug molecule in the market, you are talking about revenues, licensing deals that can go up $1 billion US. So if you invest a lot in research and science and technology, all you need is 1 or let's say 5% or even 1% of your projects to be successful. And it will more than compensate for all the money you spend on those projects that were not successful. All right? So we need our policymakers to probably consider some of these factors and how can we move science and technology forward so that we can actually have some tangible benefits in terms of economic development and social development as well, because it impacts on the social aspect of our country as well and region. Okay? Okay, so that's a very technical part, and in the meantime, we labor on. Whatever we can, whatever we do, we won't give up. We'll continue to fight and see how it ends, right? Well, first of all, I'd like to thank, again, everyone for attending. I really appreciate your presence. And just to thank some special people. My parents, my father is no longer with us, but my mom is here, and she was a stalwart. Yeah, coming from this type of environment where it was very, very economically challenging, but yet she insisted that every one of us should have a secondary school education, and it was not easy. She was a housewife, she was a cattle farmer, she was a gardener, and she was a seamstress. All of that. So life, started very, very early in the morning and ended in the dark. But she made it. We have all the thanks and appreciation. So all my work is dedicated to her and to my dad, right? And again, family. It's a big family, so I have to use a big picture. So it was 9 siblings, and as was indicated before, I didn't like school too much, and I never liked the environment. But all my thinking ability, I feel, my appreciation and thirst for knowledge came from interaction with my sibling. They were elder, and I was there absorbing when they were discussing their literature and their history and science. Everybody had a different interest, and I sort of took a little bit from all of them. So again, whatever I achieve is also because of my siblings. So my family as well, it takes often a lot of time with students, and they are very understanding. And I have to be thankful for not spending the amount of time I should have. Okay, but they are very, very understanding, thankfully. And then all the other relatives, my nieces and nephews, often I would say, okay, I need to spend more time because they're all into music now. And I really enjoy playing music with the family and the family group. But sometimes I'm unavoidably upset, right? But I'm hoping that more and more I'll be able to spend some time to play music and get back with the family often. My supervisors, I mentioned, they were my gods, right? Dr. Tonawa and Professor Umaran, they have had so much powerful impact. Whatever I did, I also dedicated them because it's because of their efforts I'm here where I am today. Right. So again, the deans and heads, deans of Science and Technology, FSA, Professor McDavid, Professor Narayan Singh, and Professor Ramnarayan, Dr. Kubun, and my heads as well that serve while I was here or since I'm here, Professor Bacon, Dr. Charan. Again, Dr. Kubun was my head as well, Professor Agard and Professor Gobin. Now, when you are busy with students, obviously you do not always meet deadlines. Right? So they have been very, very accommodating, and the dean will know if a deadline is today, mine time is 11:59. That constitutes today, right? Not 4 o'clock. And as I said, I often work with functional deadlines, not calendar deadlines. If I know you need this tomorrow, you're getting it by tomorrow. Although it might say, okay, please submit by today. But I've always had that good relationship and strong support, to get research done in an environment where you don't have resources, you don't have reagents. Sometimes you have to go to the heads of departments. And I remember talking to Dr. Kubo and sit down. I say, I need this and this. A long list. Shake your head. You say, yes. So what we get in? I say, you're getting papers. You won't remember, but I remember. And it did come eventually. It took a little while. But, it come and eventually came. Prof. Agard as well was very, very instrumental in helping me move forward. Prof. Gobin, thanks a million for all. Dr. Charan as well, if she's here, I'm not sure if she's here, but she has also been very instrumental in helping with getting research and helping with teaching and so on. Okay, I forgot to mention all the colleagues and collaborators. You all were listed before. I didn't want to be redundant. So all my colleagues in my department in chemistry and engineering, I work with people in FFA, in medical sciences. Thanks a million. I really, really appreciate it. We couldn't have achieved a fraction of what we did by ourselves. Special thanks to these very, very close and dear friends who are my closest collaborators as well. Dr. Dawn Phillip, she was my partner in crime. We're going out in the field. Unfortunately, she passed away. Professor Chedi as well. He was like a standard in terms of hard work and dedication. His life was for research. Sometimes when things are a little difficult with me, I say, I'm not thinking about Prof. Chedi. It's not really hard. When we compare the time and effort Prof. Chedi would spend in his research. Okay, and they are very, very well, they are missed a lot, and I appreciate all that they did that helped my research move forward as well. Current and past technical support staff in FFA and life sciences. I came out of FFA. My PhD was in agriculture. So I have a lot of people to thank. When I now started a project, I learned a lot of my techniques and so on with the technicians, and I'm so much appreciative of that. Isa Mohammed and Jane Awai, they were two senior technicians. So I was lucky in that lab to have two technicians. All right? And as I said, we did one microbiology lab when I was in year 1. And at that time, I wasn't completely transitioned into a good student yet. In fact, I went into the lab. I was still in the theoretical. I was enjoying science, but more theory. I went to the lab, and they told me, this is a demonstration lab. It's not been marked. What I did, I went out. That shows how bad. So when my students are delinquent and so on, I'm very, very sympathetic. And I try to convince them, I try to give them, some kind of pep talk and so on, try to pull them back. I never condemn students, even if they are bad, okay? Because that would be hypocritical, right? Yeah, so I learned a lot when I started undergrad project, actually, and then I did my master's and PhD, so the technicians were there to guide me and get me off and providing a range of support as well. Rabia Hussein was my technician when I now came into UE, in the Department of Life Sciences. And again, I was very narrow. I did PhD, but I didn't work with a whole lot of different organisms. But she had the experience, so she was there to support me and help move things forward. And again, I'm appreciative of that. Adesh Ramsubhag, again, she was for a long time. So she was always there to support If we have students in the lab, we need someone to be there to overlook, and she was always willing to help, oversee and make sure everything is well in the lab. Again, chief technicians like Karen as well, she was very, very helpful. And all the technical staff from the department. Again, students, I'm motivated by students. I'm thankful students. I learned a lot because of the students. They are the ones who drive me to learn. If I have to learn for myself, it might, it wouldn't be as effective as learning to guide a student. Postdoctoral fellows, we had a few of them, Dr. Chinnan, Dr. Ramjay, some visiting scientists, the plant microbe team, Ministries of Agriculture and Health, all the farmers that we have been working with over the years, and our funding agencies. Without some of these external funding agencies, and within campus as well, several funds we access, the ITT, RDI fund. Stacy has been very, very supportive as well to help move things forward, to access funding, and several external funding agencies as well. So thanks to them, and thanks to you all. And now we could probably take some questions.
Adesh, I must congratulate you on a brilliant lecture. Well done.
Thank you, Dean. Thank you, Deputy Principal. Way back.
Yeah, I must say I knew a bit about your work, but I wasn't really aware that you were doing so much. And it's really difficult to imagine someone involved in so much good research and so much applicable research. So well done. I think you thoroughly deserve that SAPGA award, plus the money that went with it. I know it's a lot of money. As well as the VC's award for excellence in research. And of course, gaining your professorship at a fairly young age. In fact, as you know, I told you should have applied years before. Then you actually did. So well done. As you are aware, I have a deep interest in plastic pollution. And I've been reading a bit about these plastic degrading bacteria. Could you share some information on whether that might be a possible solution to our pollution problem?
Yeah, so plastic degrading bacteria have been found. They apparently are widespread. We have started a project, but we didn't complete it as yet. That was before the pandemic, so it was abandoned. So students went their way. I'm pretty sure that we do have plastic degrading bacteria, because of the diversity we have. But whether or not it is a solution, I don't think the technology has evolved to that scale as yet. Like for instance, we have been hearing about biofuels from bacteria for decades. It's just not commercially viable. It will take too much resources and efforts to apply the technology to get any substantial benefits. So I don't think we are there yet with plastic, because the amount of plastic that is generated, to get that to be degraded by microbial systems, you have to probably develop some bioreactors to enhance the rate of degradation. That will require a lot of energy. So the trade-off would not probably worth it. But from academic standpoint, yes, there's a lot of interest.
Yeah. Thank you.
Good afternoon and congratulations.
Thanks.
I recently did a scan, a biomedical scan using bioresonance, RIFE technology. Is the university aware of this type of technology? It's to detect and to kill bacteria, viruses, and detect illness.
Yeah, so I've heard about it, but unfortunately we have no interest, or I don't say interest, no activity right now in that technology. It's something we'll be very much interested. Yes. Excellent, thanks.
Hi, good day, Prof. I was wondering, what is the biggest barrier in your way to getting funding for research that you'd say in your experience?
Biggest barrier is there's none available. I'm going to say in campus we do have, we are fortunate, there is a CRP grant that we consider seed funding, right? So this is accessible to all MPhil and PhD students. It's not a lot, but nevertheless, it can get you started. And as I say, yes, we need funding to do high-level research. But if you are a true researcher, then you'll live within your means. We can do simple things in the lab that requires very little resource. You could be innovative, come up with your own idea, your own solutions. For instance, if we grow bacteria, we use media, right? Yes, we use commercial media, but we can make media in some cases, or we can innovate, develop new media. So in that sense, you have to be innovative to come up with these solutions. You cannot depend on using systems that may be developed in other places. You have to adapt to your environment.
A related question. Thanks for your talk, Adesh. I understand we're all working with limited funding. I think we can all relate to that. But it still seems like a really unfortunate situation that you can have a successful research track that stops while you're still active on several other research tracks that maybe aren't as successful. I wonder what your recommendation would be to the people providing funding to provide a solution, so at least when you've got a hot potato, you can run with it,
Yeah, well, fortunately, we do have ITT fund and RDI fund. So if you have something that has that potential, I think they would be able to provide some funding. But you have to demonstrate. It has to be a little bit advanced. So there's a lot of projects that stops before we can demonstrate the potential impact that it may have. And we are trapped, right? But again, as I see it, there are different ways to look at it. I see the personal development in the students as well, and that is a satisfaction by itself. So at least, yes, we didn't achieve what we wanted or we initially intended, but the student developed. Students grows personally, intellectually, and hopefully now the students can go in another institute or maybe right here and continue to work. So that is how I also look at it. It's easy to become frustrated.
Thank you very much, Professor Ramsubhag. It was really very enjoyable to listen to your presentation. And I would like to ask you, since you mentioned that you take a great pride of supervising students, is there any criteria? How do you select your students?
Again, if I apply criteria, I'll be hypocritical. So I don't apply. I believe everyone has the ability. Sometimes people may lack confidence, or they may have some kind of mental block. So my approach now is to try to find what switch to turn off or turn on. I really believe everyone has that ability, and it has been demonstrated. Some of my best students, most accomplished students, would not have even gotten to do postgraduate studies, but they have excelled and became world class. So I am very strongly opinionated about that one.
Am I up? Adesh, the number of known described named species of everything in the world is about 1.5 million. The various estimates of the true total arrived at by extrapolation from known facts usually center around 4 to 10 million. Now you've given us an estimate of 3 trillion. Is this a fantasy? How was that arrived at?
Right. So the whole question of speciation is a big argument by itself, right? How do you define a species? Is it interbreeding population, or does it have to have some kind of biochemical divergence or genetic divergence? And this is applied to different schools of biology. In microbiology, we use sequence divergence, ribosomal sequences. So generally, if there is certain levels of similarity in that gene, we could assign organisms to species or genera. So yes, it's not practical to name 1 trillion species. So what we do, we call them operational taxonomic units, or amplicon sequence variants. So we have an alternate form where not having to name them, but we recognize the record, we keep it in a database, and we can match new organism, new sequences to the database entry. So we use a lot of bioinformatic tools to manage and catalog this level of diversity. It cannot be done manually.
Good evening, Professor. Thank
Hi.
you very much for your inspiring presentation.
Thank you.
So this is all new to me. I am definitely not in this field. I'm basically in early childhood. So this is right up my alley. So you made mention in terms of our water and your demonstration of, shaking the lettuce and, we are consuming all these pathogens, all these microbiomes within our system. How is it, let me see, are you in the rooms of policy makers in terms of how we draft our policies in terms of our health system? Because in terms of European countries, they do not purchase certain fertilizers from the US because they look at their health for the population of their countries. And we, as a society, we spend money on frivolous things. We import a lot of things that are not economical and health conscious moves that we make on us as a nation. How are we to mitigate those factors as, speaking to our Ministry of Health in terms of what we do? What are we using in our fertilizers, and not just shaking our waters? I'm curious to find out about these things because, we have our generation coming up. What are we doing to our generation?
Okay, thanks for the question. So first of all, I think we need to make decisions based on data. We are quick in the Caribbean to import systems and see if they are, apply them wholesale. All right. So we need to really collect the data, establish the risks. We just cannot go blindly and say, okay, the risk is the same here than in other countries. It might be higher or it might be lower. Sometimes the microbes can be protective as well. We might have pathogens present, but because we have so many other diverse types, they may be countering and keeping that pathogen in check. We have certain levels of the organisms that need to be present to cause disease. So maybe that is protecting us. We don't know. So this is something that we really need to really get some data on. And this is what I was talking about initially, this research and innovation. This is where it comes in. To go plow deeper into systems, investigate the environment. Don't apply things wholesale. That it may not work. I've seen that over and over. So for instance, looking at indicator organisms for fecal contamination, we use a bacteria to do it, right? So we use bacterial species. So E. coli is known as an indicator organism for fecal contamination. But E. coli in a tropical environment lives happily in the environment. So if we detect E. coli in our food, then we cannot say for sure it is fecal contamination occurring. But in temperate countries, it's more likely to be, right? So again, in terms of the pathogens specifically, I detected that we detect frequently DNA, but again, we don't understand the dynamics in a tropical environment. Maybe there may be a new species. It has a similar feature, but may not be pathogenic, or it could be more pathogenic. So we have a lot of unknowns. And our society is constructed like that. We accept things blindly. Maybe it's a consequence of colonialism. We are trained to just accept everything that is shown to us. But I believe we need to collect our own data and do our own analysis and come up with our own solutions. And that is the start before we can implement policies.
Good evening, Professor. It's been a while. Would you say that independent research could be an avenue where further research can be done within the Caribbean biosphere?
Independent research in terms of
microbiology.
Independent individual or institution?
Yes, individual. Like say if I myself wanted to do my own research independent of any sort of institute or institution.
Yeah, once you can afford it, it's fine. It's welcome. No, I think you can start. As I said, there is a way. Whatever resources you have, you just adjust to suit. You may not be able to do transcriptomic research or metagenomics, but you could do isolations. You need simple equipment. You can modify and develop a sterile chamber, for instance. And you could screen organisms. One of the reasons why we started work on antibiotics is that it's easy to screen. We don't need any big equipment. We just culture organisms on a plate, put the pathogen, and put our environmental organism and see if there's inhibition. You can do that at home. But then, when I say at home, in a home lab, not in your bedroom, right? And obviously, you need to have some safety guidelines and safety protocols and so on. Hopefully, that is considered. But yes, you can do it. But the problem is when you get an active organism, what do you do with it now? This is where you need the more advanced capabilities. But certain applications, I think it might be possible, depending on what your interest is.
All right, thank you very much.
One more? Okay, I'm told one more question.
Good evening, Dr. Ramsubhag. Trisha Singh from Kuriri. Nice to see you again. Thank you so much for the presentation and sharing your journey. So I have been practicing microbiology for the past 18 years in a more or less commercial setting. So I find my pet peeve is I find locally there's a lot of emphasis on the food and beverage sector because of course, consumption and requirements for exportation. For the environmental microbiologists, such as myself, what recommendation do you have? Yes, we know funding is an issue, and that most probably is going to be like that for quite some time. But how do we get a shift in terms of focus on the beneficial types of bacteria for bioremediation and all these other hosts of beneficial traits? Can biopiracy be switched, for example, instead of thinking of it as piracy, can we move to a model of collaboration with these foreign agencies? Thank you.
Yes, again, it comes up to the argument I was making in terms of culture. We don't appreciate knowledge. We don't invest enough on innovation and research. And that is the root of it. So we have a lot of problems. We have a lot of solutions. But the mechanisms are not there to harness those potential solutions. Collaboration is probably one option. But then if I'm a multi-billion-dollar company, and I could get the technology by going around you, all right? And I just have to walk and collect sample. A speck is sufficient. Yeah. We could put it on a shoe below here. Nobody will check on the issue, right? So that's the problem. Yes, I think collaboration could be useful, but you have to have your agreements and so on in place. sharing of benefits. So in terms of our drug development research, if we were to license that or sell it early on, we get very, very little. But the big pharma companies, they want you to take it as close to possible as market. But if it goes into, let's say, third phase clinical trial, they are willing to give you $1 billion. But they wouldn't give you a few $1 million early on. So that's basically the challenge we have. Okay, thanks everyone again.
So congratulations and thank you very much, Professor Ramsubhag, for that intellectually stimulating, insightful, and very honest and sincere presentation. And really, your honesty really has inspired all of us, especially your academia, your journey into academia. I don't think many of us who have gone through academia may not have been so honest to express what you have said today. And I think we should all put our hands together. Yeah. At 5:20 today, I was heading towards the campus and I saw Professor Ramsubhag heading in another direction. In his, and this is, I'm not making this up, in his trademark sweater. And I'm blowing my horn. We stopped at the corner, I'm blowing my horn, and he's just whistling. I say that in the context of how you made your presentation. Very calm, cool, collected. Once again, a round of applause. And it really indicates, the high quality of presentation, the high quality of presentations. At this point, I'd like to invite both the campus principal and Professor Ramsubhag on stage, and I'll ask campus principal to present a token of appreciation from our open lecture committee to Professor Ramsubhag. Please. Campus principal. Would you like? Thank you, Campus Principal and Professor Ramsubhag. As we near the end of our proceedings, I'd like to invite Ms. Suming Nathaniel, currently Professor Ramsubhag's PhD student, to deliver the vote of thanks. Ms. Nathaniel was previously a microbiology student of both Professor Ramsubhag and the late Professor Dave Chady. She is currently the Senior Technologist and Sequencing Team Lead at the Caribbean Public Health Agency. It is my pleasure to invite Ms. Nathaniel to deliver the vote of thanks.
Ladies and gentlemen, as we end this stimulating and enlightening inaugural lecture, I have the privilege to extend a heartfelt vote of thanks to everyone who has contributed to making this evening a resounding success. First and foremost, I would like to express our gratitude to Professor Derek Chady, Chair of the Open Lectures Committee, for graciously presiding over this evening's event. I'm profoundly grateful to to Professor Adesh Ramsubhag for his exceptional presentation on the local microbial landscape, the challenges it presents, and the remarkable biotechnological potential it holds for the Caribbean. Professor Ramsubhag's comprehensive exploration of this topic, combined with his passion and expertise, has not only inspired and enlightened us, but has also underscored the significance of understanding and harnessing the power of these microscopic organisms for the betterment of our region. As the featured presenter, Professor Ramsubhag has truly captivated our attention with his depth of knowledge and engaging delivery, demonstrating the vital role of microbiology and biotechnology in addressing some of the most pressing issues in the Caribbean, from the environmental sustainability and human health to agricultural productivity and industrial applications. His presentation has revealed the myriad of opportunities that lie within our local microbial landscape. We are indeed fortunate to have had the privilege of learning from Professor Ramsubhag this evening, as he has opened our eyes to new possibilities, ignited our curiosity, and instilled in us a sense of responsibility to further explore and unlock the untapped potential of our region's microbial treasures. Let's have a round of applause for Professor Ramsubhag. We are indebted to Professor Rosemary Ballantyne, Pro Vice Chancellor and Campus Principal, for her opening remarks and unwavering support for this prestigious lecture series. Our thanks also go to Dr. Brian Coburn, Dean of the Faculty of Science and Technology, for introducing our esteemed speaker and for his leadership in fostering academic excellence. I would like to express our appreciation to our colleagues at the UWI St. Augustine Campus, specially invited guests, and members of the media whose presence has made this event even more significant. As a PhD candidate at the University of the West Indies, I am personally grateful for the opportunities that this institution offers to young researchers, and I extend my heartfelt thanks on behalf of my fellow students as well. Finally, I would like to acknowledge the efforts of the Marketing and Communications Office for their hard work in organizing and promoting this event. Let me once again express our deepest gratitude to everyone who has made this evening a memorable one. We look forward to many more such thought-provoking and inspiring lectures in the future. Thank you and have a wonderful evening.
As we come to the end of the formal program, I once again want to thank our featured speaker, Professor Adesh Ramsubhag, for that excellent presentation. Before I bring this session to a close, I'd like to invite all to join us in the foyer. The ushers would guide you for refreshments. A reminder to Professor Ramsubhag and family, campus principal, deputy campus principal, and dean to remain in the auditorium for a photo shoot. And a very pleasant good evening to everyone, and thank you all very much. I now formally bring this session to a close.